It's my pleasure to be here today and to talk about where we see some of the future of genomics heading. So it's a little bit of crystal ball gazing, and I guess there'll probably be some differences in opinion in the audience. So the first question I have is there anything that's 300,000 times cheaper now than it was in the year 2000? So when I asked my dad this question, he said, the price of milk. So, maybe it is in England, I don't know. Anyway, I was thinking, well, computers, they're cheaper than they were in the year 2000, but not by that much, possibly by about 5 times if you compare like for like. So what about flying? I do a bit of flying every now and again. If you go with some of the cheaper airlines, such as Jetstar in Australia or EasyJet over here, then it could be up to 3 times cheaper if you're lucky. What about newspaper? We all used to buy newspapers, now most of us download it. Well, you still have to pay for the cost of download, so maybe 2,000 times cheaper. So, what is 300,000 times cheaper is sequencing technology. And it's really reduced in price phenomenally from when we started. So when the first human genome was sequenced, it cost somewhere in the region of $300 million. And it's gone down really quite dramatically since then. 2010, you could get a sequence done for $5,000. I read in Scientific American in January of this year that by the end of the year, a $1,000 genome will be a reality. So 300,000 times cheaper is looking very achievable. So what is sequencing and what does it mean? Well, DNA, obviously, it's shared by all organisms. So regardless of whether you're looking at Drosophila frogs, any type of fruit, plants, or dairy cows, they all share the same DNA. So we can apply the same technology. We can use the sequencing technology regardless of the species. And I think this is a major way that agriculture, and in particular the dairy industry, has really leveraged off the investment that's been made in human medical research. So we're some of the beneficiaries, I suppose, of huge investments that have been made elsewhere. And we've really run with it in the dairy industry. And we're already capturing the benefits through genomic selection, and we will do in the future with sequencing technology. So the ways in which that we use this has been mentioned by other speakers this morning, is to improve the profitability of dairy cattle. So improve fertility, production, feed conversion efficiency, survival, disease resistance, type traits, you name it, we can do it. Also decreasing environmental impacts such as methane emissions in milk and so on. So how do we do it? Or what does the sequencing technology tell us? Well, basically, sequencing technology is a way of reading each little bit of the genetic code. So all those A's, C's, G's, and T's will actually read in a bull sequence. So you end up with, if you like, a whole sequence of letters. The bits that tell us or that are most useful to us are what we call SNPs, or single nucleotide polymorphisms. And these are pretty much spelling mistakes in the genetic code or differences between animals. So once we've found the differences between animals, we can use this to understand the genetic control of the traits, and obviously use this to predict breeding values for bulls and cows and to select on these. So this has got a great potential, and we're already seeing it, to select bulls and cows for breeding early in life and accelerate the rate of genetic gain. So sequence data is definitely in the realm of the future, but we're already using it in the form of SNP chips and genotype data right now. What we are doing in preparation for the future is with sequencing of key ancestors. So there is a project that's going on called the 1,000 Bull Genomes Project, and this includes any groups that have sequenced or have whole genome sequences of bulls or cows of any breed of cattle. So this is a slightly outdated version. So when I put this slide together, we had 151 sequences. The number is currently 242, of which we've done 139 at DPI. in Melbourne. So there are a mixture of different breeds. There's Holstein, Fleckvieh, Jersey, Reds, Angus, and there will be other breeds. We're well on the way to achieving our target of 400 by the end of the year. And I must say, this is absolutely a global effort. There's many different participants that I'm sure are here in this room and that will be attending the conference later in the week. So the bulls that we've sequenced, well, they are the granddaddies of the Holstein breed, for instance. So on the left here, I've got two photos of bulls that I would consider to be very much ancestors of the current dairy cows. So the top picture is Glenafton Enhancer. He's been sequenced, and so has Eastland Cash, the picture below. And you can see there's other very well-known bull names there. And this is obviously only a snapshot of the bulls that we have sequenced already. So that fold coverage number tells us basically how many times we've read the sequence, just to make sure that we've actually got it right. So for bulls like Starbuck, we've gone to very deep level of sequencing, 30 times. How are we going to use this information? Well, the idea is that if you have the sequences of these key ancestor bulls, and if you have SNP data or genotypes on the descendants of those bulls, then you can fill in the gaps. So you end up with what we would call imputed sequence on those descendants of those bulls. The reason why we're doing this is because if we're able to capture the differences or all the sort of key mutations that are responsible for variation in a trait, then we can increase the reliabilities. So the goal is to try and get the reliability of genomic selection to the kind of reliabilities that we're used to with progeny testing. So from 0.6 to, it would be nice to get to the 0.8 level. We also recognize that there are other ways to increase the reliability of genomic selection, such as having larger reference populations. And again, there's international collaboration that's going on to bring together genotypes and phenotypes from lots of different countries. So we'll end up with international reference populations. There's also the opportunity, of course, to use information from other breeds. And increasingly, I think we'll see a move to using cows in the reference population as well. And that's basically because we'll have fewer bulls coming through the progeny test pipeline, and we'll need to update our reference population in another way. So, to cows, what do we have available? For the bovine SNP chips that are available are the 7,000, 50,000 and 800,000 SNP chips. And there are, of course, other chips available for different species. I'm quite sure that there'll be other flavors of SNP chips that we'll have in the future, and perhaps even different ways of capturing genetic information. But it's probably outside the scope of my talk to discuss those. I think one thing that we may very well see in the future, I wouldn't want to put a date on it, but it would be that all cows and bulls in the key dairy nations will be either genotyped or, in the case of bulls, possibly sequenced. What this will enable us to do at both the cow and bull level is to identify the best individuals and increase the rate of genetic gain. At the cow level, it will help us, especially for those of us in pasture-based systems, to unambiguously work out what the sire and dam of a particular calf is. So really useful there. It'll help us to select the best heifers as replacements and a way of controlling inbreeding or managing matings. So I think this is probably a pretty well-known sort of slide for most places around the world. And it shows the advantage of progeny testing compared to genomic selection. And of course, as soon as you have DNA, you know what the genetic merit of a bull is, and you can save, we think, around about 4 years in terms of faster rate of genetic improvement. So one thing I think is a way that we can use this technology even further is we're obviously getting thousands of bull calves that are being genotyped worldwide, and they'd have a reliability of around about 60% at birth. And we can use this information both within country and across country if we're prepared to exchange the genotypes of those young bulls. So for a country such as Australia, there might be bulls that are born and genotyped in another country that might be the best for that sort of situation. And of course, the same goes for no matter which country you're from. I also think, and I think it's been mentioned already, that a shorter generation interval means that you have a really rapid turnover of bulls. So I think we're probably less likely to see these sorts of breed icons that keep going for year upon year. And you may have a situation where a bull would be high impact for a year or two before he's replaced by a whole bunch of a lot better bulls, especially with globalization. And we're probably going to see a reduction in the number of bulls progeny tested as the confidence in genomic selection grows. So one thing I think that we'll see a lot more of is the use of computerized mating plans. Now I see this as coming from the fact that if you've got a really high turnover of bulls, then where some farmers have had a very good storage capacity for bull pedigrees in the past, it might become a lot more difficult to actually remember what sire and dam that particular bull was by, and therefore which is the right cow to mate that bull to. So in terms of inbreeding, I think that we'll see this sort of technology really starting to fly in the future. And the other great thing about using genomics is that we're able to calculate the relationship between individuals in a much more precise way than we've been able to do before. So I have a little example of how we can do this. So these are 5 ET sisters from a herd in Australia. They could just as well be 5 ET full brothers. The heifer in the middle, the black heifer, she is sharing genomically more of her genome is shared with the heifer with a white blaze down her face than the darker heifer that looks a little bit like her. So looks can be deceiving, I guess. So if you were to use just pedigree information, then you wouldn't be able to distinguish between those heifers, and you'd just be able to say that on average they would share half their genome. And using this genotype data, we can actually distinguish between them and say, no, this heifer actually has inherited more similar genome to the other heifer compared to a different one. And obviously, as with bulls, we can use this information to work out which is the best one at a very early stage. So, how you might use this information. I've just got a little example of from the same herd that those 5 ET sisters came from. We also ran through a mating program where we selected 10 genomically tested sires. And we worked out their pedigree relationships, which is the little chart on the left-hand side, and also their genomic relationships. And you probably can't quite see it in the image up there, but with the genomics, we're able to decipher between those relationships in a much better way. And this means that we can actually control inbreeding to a greater degree using that genomic tool and the pedigree tool. So for the scenarios that we ran with this particular herd, we can save 2.5% inbreeding compared to only 1.4% when we use pedigree. And of course, this has a value to it. You know, inbreeding results in depressed production, reduced fertility, and so on, and that has an economic value. So in Australia, we reckon that it's worth about $50 per lifetime, which translates to 39 euros. So that's 39 euros per mating through this sort of strategy. In the future, will we still need to record data? I think Dana did a very nice presentation on lots of different reasons of why phenotype data is important and why we should record it. And Mike Coffey, one of my previous colleagues at SAC in Edinburgh, is trying to coin this phrase of, in the age of the genotype, the phenotype is king. And I think it's absolutely right that the genotype information is pretty useless without the phenotypes to go with them. So why do we need it in the case of genomics? Well, to make sure that the associations between the markers and the current population remain relevant, so our prediction equations remain accurate. And I think there has been a history of genetic evaluations really being a byproduct of data that's recorded for other purposes, such as management. And we do face challenges going into the future of how to capture data from new types of systems, such as inline recording. I also wonder what will happen to data that we've traditionally collected as part of progeny testing schemes, such as type data. So, for example, in Australia, the progeny testing organisations actually record the type data from herds that participate in progeny testing. So is there a role for industry resource herds to make sure that we still get this information in the future? We also have a potential to, and I think this is actually one of the really great strengths of genomic information, is for new traits, such as feed conversion efficiency. So measuring feed intake is very expensive and you need specialist type equipment. So genomics actually enables you to use information from, say, resource populations, calculate the genomic prediction equation, and then apply this to animals that have got genotypes but don't have the phenotypes. The same goes for methane emissions and really good fertility measures that are possibly a little bit too expensive to measure on a large scale. So just a little bit of what we've done in the area of feed conversion efficiency in Australia and New Zealand. We've had, we've run a trial where we had 1,000 heifers from Australia and 1,000 from New Zealand. We measured residual feed intake. Residual feed intake is what we predict a heifer would eat minus her actual intake. And we worked out that the heritability of this trait was around about 0.25. So there is genetic variation there. And we also calculated that the accuracy of genomic prediction was 0.4. So in terms of implementation, 0.4 is perhaps a little bit on the low side, but we're hoping that through international collaboration that we'll be able to improve this prediction, and also getting more phenotypes on Australian animals. So, in summary, what does the future hold for genomic selection? I think higher reliabilities is one thing that we're striving for, and we'll be able to make this happen using more and more genetic markers and through that sequence data technology. I think we'll see a gradual reduction in progeny testing, but we absolutely need farm data to make sure that the genomic predictions are still relevant. I think as genomic selection or the uptake of it increases, there'll be a more rapid turnover of bulls or sires of cows and bulls, and we'll see this resulting in what I call fewer breed icons. And as a result, mating software to avoid inbreeding is, I think, going to become more important. And finally, there's new traits such as feed efficiency and better fertility measures and so on. So with that, thank you very much indeed.